turn 2. All right. So where we ended up on Monday is that we basically spent, more than half the time working on calculations for transportation. And our topic has been, both on Monday and last week before the test, we've been talking about membranes. And that's what we're going to continue on today, just talking about biological membranes, right? And since we ended our class on Monday talking about transportation of ions, the energy of it, that is, let's actually talk about how ions can go across the membrane. And so this word is cut off on the bottom for you, so it should be valamycin. All right, so ions are charged particles, and the membrane itself, remember, has nonpolar plus polar heads. So ions can't go through our membranes naturally. They just can't diffuse through because they never want to lose the water that is surrounding them, right? Because to go through here without any kind of pore, you have to go through desolvation, lose your water. For an ion, the energy is so high for that to happen, it's basically impossible. And so we have two different types of ways or ionophores to move our ions across. One is just a carrier, and it's like what it says, in that it will just carry an ion across the membrane. And the other one forms a channel or a pore, which will allow our ion to go through. Both greatly speed up. Basically, it goes from 0 speed to, thousands to hundreds of thousands of molecules a second. And the cool thing about these ionophores is that they are very specific, right? The cell, the things inside the cell, they're at very specific concentrations, right? And different ions can send different signals to your cell. So you want to be very careful about what you're transporting at any given time. So these cells have been, been molded after millions and millions of years to be so specific to only carry one type of ion. And so our example is valamycin. And what we're looking at, and I know it's a little cut off, but that's fine, is this is our potassium ion right there in the middle. And here are all the amino acids that surround it. And this cage is electron density. Just to let you know how this model was made, basically, when you do like X-ray crystallography, you only get this cage back, and then you have to use a computer program to put the amino acids in. So that's what we're looking at here. And you can see that this potassium fits perfectly into this little pocket. It has a bunch of negative oxygens. That's what the red are. The red are oxygens. And all these oxygens are arranged in such a way that only something that is positively charged and the size of potassium can go through that channel. So that's how we can be specific about what's going through our channels. We have the right amino acids who interact with the charges, and it also has to do with the size of the hole that we are making. So just our first question is just to understand, what can and cannot diffuse through the membrane, just so we have an understanding of exactly what we need a pore for and what we don't need a pore for. I believe I have, do I have this? I don't. Thought I made a poll for this. Apparently I didn't. So what I want you to just look at these molecules and just ask, do you think you need a pore or mediated transport for this, or do you think it can be non-mediated? So just take a minute and try to figure that out, and then I'll go over the answers. So all the names do go with, so that's ethanol, glycine, cholesterol, ATP. All right, so let's take a look at this. Ethanol, this one is a little surprising. That can actually go through non-mediated. So that can actually transport through the cell. And I say it's a little surprising because it does have a polar group, this OH, but it can still get through the membrane because it does have a large nonpolar section. So if you thought that one was mediated because of the OH, that would be a perfectly logical observation. But this can actually pass right through the cell, right? Glycine cannot. This one has to be mediated. Now, it's similar to the same size as ethanol, but one big distinction: glycine is charged, right? So nothing charged can go through the membrane unaided. That's like a pretty hard rule there. So since glycine is charged, it can't just go through the biological membrane. Cholesterol, well, this is all nonpolar, so that's actually non-mediated. That can just slip right in through the membrane. ATP is mediated because all the phosphates there are charges. So when thinking about, do I need a pore or do I not need a pore? Some of the things you're looking for is if it's charged or if it's very polar, or like we'll talk about the water. Water does need a pore to help with diffusion. It can go through the cell by itself, but it's super slow. So you need aquaporins, and that's because it's polar and there's no nonpolar component. Ethanol does have a pretty big nonpolar component, so that's why it's non-mediated. But as I said, if you put mediate for ethanol, totally reasonable. Any charge is mediated, no positive or negative. If a molecule is charged, it's not going through the membrane without some kind of protein to bring it through. So any ion. Basically, polar groups do not want to go through the lipid bilayer unaided as well. But there's some, but it gets a little bit tricky in that just because you have a polar group, you could have a large nonpolar group to go with it. So usually polar things don't want to go through the membrane either, but there are exceptions to that. So remember, and the reason why this is, right? If we go back to our previous slide, If I want to go through this membrane, I have to go through this whole nonpolar region before I can go to the other side. Polar things don't like going through nonpolar areas. It's possible if you have a large nonpolar area yourself. Charged things, anything with a net positive or net negative charge, absolutely will not go into the nonpolar region. It is way too energetically unfavorable for anything with a charge to go through that region. And that's why we need carriers to do that, or channels. So we're going to look at pores today. And the pores we look at are going to be, I'm going to say, variation on a theme, in that pores more or less work the same way. Their inner channels have been evolved over millions of years to be specific for one type of molecule to carry. So we're going to look at porins to begin with. And a porin is a class of proteins, right? So there's multiple porins out there. You have aquaporins, but what we're going to look first at are maltoporins. So porin is just a beta barrel. Remember, beta sheets. It's a bunch of beta sheets in a barrel. And in the middle, we have our channel, which will bring through a molecule. A maltoporin will bring through what's called maltodextrins. And it's just a sugar, basically. So it's a specific sugar, or specific sugars. And this is a homotrimer. So homo means same, right? Trimer means 3. So it's the same protein copied 3 times to make one structure. And each protein are 18 anti-parallel beta sheets. So this is an hourglass shape, the inside of it being 5 angstroms, right? And angstroms, you probably don't know how big of an angstrom is off the top of your head. So the definition of an angstrom, 1 angstrom is a carbon to hydrogen covalent bond. So very, very, very tiny distance. And so this is only 5, carbon hydrogen bond lengths wide. So it is not wide at all. It is very tight in there. And this maltoporin has a bunch of aromatic amino acids. So remember, in case you've forgotten, since it's been a long time since we talked about amino acids, W is tryptophan, Y is tyrosine, F is phenylalanine, right? And so this yellow here, we have a bunch of hydrophobic amino acids lining this channel, which makes a lot of sense, right? Because we talked about in the sugars chapter, we were looking at sugars. Sugars are just big carbon rings with OHs pointing off them, right? And so how this works is that these aromatic residues form pi stacking. If you don't know what pi stacking is, I'm going to stop sharing for a second so you can see my face. Because what pi stacking is that if I have, imagine my hand is the glucose ring, right? Pi stacking is I take another aromatic residue and you kind of just form like a little sandwich. And this is a very favorable interaction for our molecules. So what happens with our aromatic amino acids, we have our sugars, and then we have a bunch of aromatic amino acids all through the channel, helping us move through that channel, right? And so we have a cork, it's like a corkscrew that goes through here, because all of our aromatic residues are positioned in a way to corkscrew this through. So that's kind of like the path it's going through. And there are a bunch of hydrogens, OHs, on our sugars, as we learned about in the sugar chapter. So we have a bunch of side chains that can hydrogen bond with our maltoporin. This type of structure, these amino acids, allow for only maltoporins to go through, or maltodextrins to go through, right? the combination of our aromatic rings plus amino acids that can hydrogen bond with our sugar makes it so this is incredibly specific. Any questions or any clarification on the information that's presented here on our multiple porins? Right. The next transporter we're going to look at is potassium channels. And as you could guess from the name, potassium channels transport potassium, K+. The channel we're going to look at, the actual name is KCSA. And we're just looking at one channel here. There are many different potassium channels, but they basically all work the same way. Here we have a homotetramer. Again, homo is same, tetramer is 4. So it's the protein copied 4 times, making one structural unit. And they're very selective in that they only let potassium through. They don't let calcium through. They don't let sodium through, only potassium. And how this works is that the subunits kind of form like a cone almost, where they form the pore, right? So you have, it's really hard to see on these pictures, but it's the protein just kind of forming a cone with a hole in the center. Now, the pore itself changes diameter depending on where you are. So we have one part of the pore that can be up to 10 angstroms, and another part of the pore that can be 3 angstroms. And basically, how this works is that you have a bunch of oxygens. That's what all these red dots are, right? You have a bunch of oxygens surrounding the pore. And only potassium has the right size to interact with these oxygens, right? If another positive ion tried to go through this pore, since no other ion has the same atomic size as potassium, they won't be able to go through. They'll be too small to interact with the oxygens properly, or too big to physically fit through that pore, right? But potassium is the right size to go and interact with all the oxygens in this pore. And so what happens is that, let me erase this, is that these different oxygens, let me get a different color of pen so you can see it. These oxygens more or less kind of like hand off our potassium until it's finally ejected. So they are coordinated, and they're coordinated in such a way to keep the potassium moving through our channel there. How do these channels uptake their ions if they are surrounded by water? How do they overcome that, if that makes sense? Yeah, that's actually a very good question, because these ions want to be surrounded by water, right? It is very energetically unfavorable to take these ions out of water. And that's kind of what you're doing here, is that these ions are temporarily taken away from water to go through these pores. To get over the energetics of that, that's why we have all of these oxygens around here, because water can't go through the pore with these potassium. So what kind of happens, and I'm going to try to put it in a way that makes sense, right, is that potassium, is floating around in the extracellular matrix, and, it's having a good time. It's surrounded by all these water molecules. But if it comes down, if it comes near the pore entry here, I guess I should have chose green. This red thing is potassium, by the way. when it comes near the pore opening, all of these oxygens are going to be welcoming it in. It'll be attracted to want to go interact with these oxygens. And so what will happen is that the water will kind of like part away a little bit. So water will kind of move away as these oxygens up here begin to interact. And so the pore, the opening of the pore, and you can see it's kind of shown right here. All of that red that's being shown there is negative. So the top of the pore is extremely negatively charged. And so the potassium has two choices, right? If I'm positive charge, I can either interact with water, and the oxygen of water basically has a negative charge of 0.70, or I can interact with this with 4 oxygens. And that's what's being shown there. 4 oxygens, each one with a charge of -1, right? And so the potassium is going to want to interact with these oxygens, and that's how it gets rid of the water. The energy penalty it takes for getting rid of the water, it makes up for by interacting with these new negative charges, if that makes sense. I know that was kind of a long explanation, but short, my 1 to 2 sentence answer is the top of the channel is so negative that the water will be stripped away when potassium races towards it. That's the super short explanation of what's going on. All right. So let's say you had a channel for phosphate, right? Phosphate is PO4 3-. What two, I'm going to say there's 2, what 2 amino acids do you think would be at the top of our porin to try and attract that potassium ion towards us? This is a good review on amino acids because it's been a while. We've been talking about boring sugars. Well, sugars aren't boring, proteins are just cooler. So what type of amino acids, or what 2 amino acids would be at the top of a porin for phosphate? Phosphate's PO4, there we go. PO4 3-. Not L. It is K. K is lysine. L is leucine. L is nonpolar. You got half of them. What's the other one? What's the pirate's favorite amino acid? Arginine. Yep. So at the top of this porin would be R and K, right? And so for any type of porin that we want to use, the top of it is going to be extremely well, it's going to be designed to be extremely favorable to interact with whatever we want to transport. So in our previous example, we would have like asp and glutamic acid to interact with our potassium. Here, we're looking at arginine and lysine to interact with our phosphates. So for potassium channels, as we talked about, what kind of non-covalent interactions? And since we just kind of answered this, I will answer this myself. The important type of non-covalent interaction here is that you want to have a ionic interaction between positives and negatives. Note that for this channel, and basically any channel, you don't want to form covalent bonds. This is something that seems to get people confused a lot. Basically, when you're transporting something, you don't want to form a covalent bond. A covalent bond is basically forever. You are forming a physical interaction between two things. You're putting a spring between two things. An ionic interaction is that's basically magnets, right? So that's going to be the most important interaction here. So when we're talking about channels, multiporins, aquaporins, potassium channels, sodium channels, I don't care what it is, that's not going to have covalent bonds. You're looking at ionic interactions. For other channels, you can have hydrogen bonds, and you can have nonpolar interactions. Hydrophobic interactions are this, right? Nonpolar says hydrophobic. So this would be for our multiporin that we talked about. This would be for our potassium channel. So just keep that in mind, the type of interactions we have. All right. So here's something I kind of did mention, and I think that was getting to like the question that was posed. So the diameter of our potassium channel is 6 angstroms. Water is 2.75 angstroms. So it is smaller than our opening, right? So let me make a little diagram. If our opening looks like that, 6, water is basically this size. So in theory, it should be able to fit through that box, but it can't. Why can't water go through our potassium channel even though it can fit? See if you can put that into your own words there. Because water has partial charges, negative charge repulsion. So for the partial charges, that is true, but it has to go with what partial charges, right? And so that goes in with the second answer of negative charge repulsion, right? So water has a partial negative. Here we have a bunch of oxygens. So if water tried to go through that pore, it would have to interact with those oxygens, and it doesn't want to do that. So that is part of it, right? So that's one. So negative repulsion. Now, water does have partial positive on the hydrogens, right? So that could, in theory, be okay. But let's look at this, right? The reason why it's not, let me get a different color here, is that let's just imagine, right, that water was able to do this. We're hydrogen bonding to our two groups. One, this oxygen would have to interact with the other oxygens. Very unfavorable. You basically have negative charge interacting with a negative charge. That doesn't want to happen. But what also is the problem, and we talked about this for potassium, only one molecule can pass through at a time. So this water molecule would have to shed all of the other water molecules around it, right? Because water is always interacting with itself. To go through this pore, it has to be alone. That's very energetically unfavorable. For the potassium channel to do that, it made up for that penalty by interacting with the oxygens around the pore. Water can't interact with these oxygens all that well, so it can't pay that price to get rid of the water. So even though it's small enough, it's not the right charge to really fit through there. Does that make sense? Questions about that explanation? Just another way of showing you these pores are incredibly specific. All right. So let's get to a topic that's actually, I think it's really cool. We can use this idea of channels and moving ions to now understand how a nerve works, right? So neurons, they're just giant voltage gated channels. What a voltage gated channel is that it's a channel that opens with voltage, right? Voltage gated, my gates are controlled by voltages, right? So what happens when your neurons are firing, which are hopefully happening right now as you listen to me, is that neurotransmitters will be released, right? So let's say that we have neurotransmitters coming in to your neuron, right? Right here. What will happen is that these neurotransmitters cause sodium channels to open. So sodium channels open. When sodium channels open, sodium flows into the neuron. And my animation got stuck there, but usually the inside of a neuron's negative when our when our sodium goes in. This causes our cell to be positive on the inside. This has a lot of different effects. One effect is that other sodium channels next to it will open up. So if one sodium channel opens up, its neighbors will open up, channel opens. And so all along your neuron, the negative charge that was on the inside will turn to positive, right? Now, we don't want the inside of our neuron to remain positive, because if our inside of our neurons remain positive, we can never use that neuron again. The way a neuron works is that the inside has to be negative. So after our inside becomes positive, which we will call local depolarization. So we have a local change of polarity. This opens up potassium channels in the same location that our sodium channels were in. Now, once our potassium channels are open, so blue is sodium. Let's do dark brown for potassium. Our potassium flows out. And so they flow out. And what happens is called hyperpolarization. The cell becomes even more negative than when it started. So sodium channels open, sodium flow in, neuron becomes positive, right? This causes potassium channels to open, potassium flows out of the neuron goes back negative and becomes really negative. This hyperpolarization will cause sodium and potassium channels to close, right? And so we have restored locally the negative charge inside the cell. And this will go on throughout your neuron, where sodium flows in, becomes positive. This opens up its neighbor sodium channel and also opens up a potassium channel. Potassium channel opens, potassium flows out, right? Inside of cell becomes negative again. Both channels close, continue on. And that is how a neuron works. The graphs that are being shown here, let me redo the time. So the x-axis is time in milliseconds. So that's 1, 2, 3, 4 time milliseconds. And you can see here, this permeability is basically telling you when the channels are open. So first our potassium channel opens, and after that first our sodium channel opens, then our potassium channel opens. Here is our membrane potential. At time 0, we are -60, right? And the scale of the x-axis is the same as well. So that's x-axis, that's 1, 2, 3, 4 milliseconds. So we go really positive, which is at the same time point that our sodium is open, then our potassium, our potassium opens up and we come really negative, and then over time we'll go back to baseline at -60. So is there any question about the explanation of how your neurons can propagate a signal? How do neurotransmitters start the opening? So I would guess, I haven't looked into this, but the way these things usually work is that this channel has a binding site for that neurotransmitter. And so once that neurotransmitter finds that binding site, it's going to cause that channel to open up. That would be my guess, but I have not looked into that specifically. But that's usually how these things work, is that the small molecule will find the protein it has to interact with. It will bind to that protein. That will cause a conformational change. Here, that conformational change will cause a sodium to open up. So when Na flows in, the inside becomes really positive, causing potassium channels to open, allowing the potassium to go outside the cell. Yep. Wouldn't that be energetically unfavorable? You mean a conformational change of your channel? Not necessarily, right? Because remember when we talked about hemoglobin? When hemoglobin binds oxygen, it goes through a conformational change. It goes from the T state to the R state. And the same thing is probably going on here. Neurotransmitter binds. We go from the closed state to the open state. That would be my guess. That's usually how these systems work. Again, I haven't looked up this specific one to know exactly what the neurotransmitter is doing to that channel, but that would be my best educated guess. Usually for the neuron, it just happens once per signal. It's just the animation. In the video you're supposed to watch, I think the animation goes the whole time. For some reason, my animation like stopped. Oh, there it goes. So let me get rid of my and it stopped again. But basically what you should be seeing, there it goes, is that the negative charge is kind of, or the positive charge inside the cell just kind of continues on. So that animation is showing you what's going on here for that neuron. And then when it gets to the end, it will activate more neurotransmitters to be shot off to the next neuron. So we can propagate this whole signal throughout the cell. So basically, the way that your brain talks to the rest of your body is just through electricity. We are bioelectric computers, more or less. How come the whole neuron doesn't become positive when Na flows in? Because sodium is so small compared to a cell that it only is able to make a small localized region positive, right? It's not like, well, I guess, let me explain this better. Like, for example, maybe only that sodium channel is open. So sodium just flies in right here, right? The rest of the cell hasn't been affected, right? Because there's no sodium there yet. So the rest of that cell will remain overall negative charge, while this one local area becomes positive. And to prevent the whole cell from being positive, which would ruin our whole action potential, we just open up potassium and we let potassium flow out, and that will return the inside of our cell negative. What's the electric part? Yeah, so inside the cell is negative. Sodium flows in, changes it to positive. Potassium flows out, changes it back to negative. These things are charged. Does that make sense? Or any other questions go along with this action potential? And keep in mind, when you are breathing, when you are typing, when you're doing anything, this is going on in your body. Every time you move, you are sending these signals, and this is going on through every one of your neurons. Make sure you eat your bananas, get that potassium. Or I'm sure there's other things to get potassium. So bananas are just your tastiest option, in my opinion. So we already answered this a lot in the last couple of slides. How does the nervous system rely on voltage-gated channels? That's the whole sodium potassium thing. So I'm not going to just repeat what I just said there. Use this question when you're going back to studying the test to make sure you know how the neuron works, right? Without looking at your notes, to know if you're going to do well on this test, you should be able to explain to me how the neuron propagates the signal using these sodium potassium channels. If you eat enough bananas, you can die from radiation poisoning. If you eat nothing but bananas, you're going to be dying of other things before radiation poisoning. But so yes, that can be, I didn't know about radiation poisoning, but you should eat a balanced diet. Okay. So what we're going to look at now is how our potassium channel actually works inside the neuron, right? So how do our potassium channels open up once the inside of our cell becomes positive? So now we're going to look into biochemistry to figure out, how does a system work inside of a cell? How does life work using biochemistry? Okay. So as we talked about at the beginning, sodium channels open up. Sodium flows in. The inside becomes positively charged. Now we'll open up our potassium channel. How's that happen? Open. First, let's actually take a look at this channel. This channel is made out of 6 different helixes. And this picture right here on the bottom left is a little confusing when you first look at it. So let's take a look at this picture at the right first. These helixes are like in a cone, like we talked about at the beginning of the class. And inside the center right here is where we are going to let through potassium. This picture on the left, the helixes aren't in the cone shape because they're only there to show you what's happening. So to better understand this, imagine that I took all of these and kind of put it like in a cone to make it look like that. So that's the big difference between these two pictures. Okay. But the one on the left is very good for understanding what's happening. So when the inside of our cell becomes positively charged, on our 4th helix, there are 4 conserved arginines. Arginines are positively charged. So if the inside becomes positive, these arginines want to move away from that. So our helix S4 is going to move away from the inside of the cell and move towards the outside of the cell. When we move S4 towards the outside of the cell, our pore opens up. So the pore, so all of these helixes are connected. So S4 moves towards the outside of the cell. That will shift the whole protein so our pore opens up. When our pore opens up, potassium can escape. When our cell becomes negative again, the inside of our cell, the arginine will then be attracted towards this, and it will move back towards the cytoplasm, closing up our pore. So that's mainly how the pore opens up and how it closes. It's all because of 4 conserved arginines. If you mutated these arginines away, that is, you changed them into like asparagines, that neuron would no longer work. it would no longer open the potassium channel, and you would never propagate a signal. Now, that's one way to open it. And we talked about one way to close it. Now, there is another way that this channel gets closed, right? Because the whole idea of this channel is to let potassium out just enough that we change our cell back to negative. You basically don't want to just throw out all your potassium at once. We just want to throw out enough for the inside to become negative. So these channels have a safety feature built in. It's kind of cut off here. So the squigglies, it's a squiggly and then a ball. At the end terminus of this channel, we have what's called an inactivation ball, right? And what this does is when the pore is opened, the inactivation ball swings in into the pore and physically plugs the hole. It's a very cool idea, but imagine that like you have the swinging pendulum and ball in the cytoplasm. When your pore opens up, this kind of just swings itself right into the hole, blocking any more potassium from leaving, right? And the way it enters this channel is that this ball probably unfolds. Now, we don't actually know exactly what's happening because we can't see on that scale, but we do know that at the very bottom of this channel, there's hydrophobic residues. And at the inactivation ball, we have some hydrophobic residues. So the idea is that the inactivation ball unfolds a little bit. It forms hydrophobic interactions with the pore. And these hydrophobic interactions, along with the inactivation ball, block anything from entering or leaving the pore. And when we look, if you were to look at other channels like sodium channels, calcium channels, these channels kind of work the same way that potassium channels do. It's kind of like the same idea. All right. So with all that information, is there any questions about what I went over for our neurons, our potassium channels, anything? Hey, I'm glad that helps with other classes and understanding cardiac medicine. Biochemistry can be really cool. That's why I got into it. I got into biochemistry because I loved how it explains like, why are you alive? And why do things around you make you stop being alive? And what systems do you need to be alive on a very fundamental level? Right. So was it 2:48? Where are we at? Okay, so I will leave aquaporins until Friday because I don't have enough time to go through another pore. But on Friday, we're going to look at how water goes through the cell. And we're also going to look at how glucose goes through the cell for when you eat a meal. Other than that, same old spiel. If you have any questions, please feel free to shoot me an email and I'll answer those. I will put homework up for this section about pores. Otherwise, I will see people Friday. Have a good rest of your day.